sometimes the mechanisms that go through and read your genes, sometimes it just throws out exons that it doesn't want, or pulls in new ones, or just reads it in different ways. So that's another type of variation, and that actually happens. That's not necessarily based on mutation. All right, so evolution and inheritance. Now that we've talked about that, so here's evolution. I fixed this for accuracy. The ocean started rusting about 4,000 years ago, and then humans. Dinosaurs are about 200. Yeah, and dinosaurs are 250. So going back to chromosomes. So I mentioned before. So again, the typical thinking, I believe, is generally, okay, you get one chromosome from the mother, one chromosome from the father, now you got one of each. Yes, that's true, but because of chromosomal crossover, you're not getting this chromosome or this chromosome, you're getting a blend of these two. This helps in a lot of ways as far as getting rid of those mutations that have already caused problems. So there's something generally like that, but with a lot more crossovers than that's showing.
How are babies made?
Fucking. Sometimes. Yeah. Right. And sometimes dinosaurs are involved, I forgot, but I don't want to step on any toes. So with that, you're getting chunks of mutation that come through all at the same time, maternally or paternally. And these chunks are what's called haplotypes. So you don't just get one mutation at any given spot, you get a group of them based on where those crossovers occurred, and they get inherited together. And these can be served as markers of traits. You can also trace population dynamics with them. It basically is, yeah, that's the same thing as what I showed in the previous slide, pretty much. And then you can do these fancy linkage disequilibrium maps, which show the likelihood of, once you have several genome sequences, you just put them all together, and you can calculate the likelihood of, if someone has this variation here, what's the chance they have this one here? And for whoever the fuck this was, pretty high. And you can do this across large swaths of the genome, and then you get what are called haplotype blocks, but whatever. But this leads to a few things, like one is a difficulty in determining whether is this mutation the one that's causing a disease, or is it this one that always gets inherited with it. But it's a hard thing to determine sometimes. But you can also use them as markers, conversely. If it's easier to find one than the other, you can at least have a bunch and look for any of those and correlate a pretty good likelihood that someone with any of them, even if it's not the one that's causing the problem, that they might have that problem. These can be done with thousands of mutations, not just a few. And using these, there have been various studies done. This one is using Y haplogroups because Y chromosomes are only paternally inherited, which generally shows pretty good support of the out-of-Africa hypothesis. Much of the genetic record has been pretty solid about backing up the paleontological record as far as what people think goes on, but there are still arguments between the two camps about when exactly those things happened. TYA means thousands of years ago. I think this actually came from a mitochondrial study, which is only maternally inherited. But again, shows basically the same thing with a differently shaped map of the world. All right, how do we know all this? We use genomic sequencing. there's a lot of types of genomic sequencing. There's Sanger sequencing is the big guns and really expensive. And then the stuff that's really cheap these days is high throughput. There's a bunch of different things, types of high throughput sequencing people can do. There's the whole genome, which is just you sequence everything. Transcriptome and exome save a lot of money because you're just sequencing the areas that are thought to be relevant to actual function. And then you got these other kinds that I just listed because. And then you can get your personal sequence, which you can combine all of those to figure a thing out. Sanger sequencing, you're basically chopping the DNA up into pieces, and then depending on the weight of those pieces, it determines which thing. It's highly accurate, it's really slow, it's really expensive. The high throughput sequence, on the other hand, cheats a lot and uses fancy computer algorithms. First it goes into a sonifier and uses sound to break the shit out of DNA, so it's all these little ass fragments. Then it gets clumped into vectors attached to like bacterial, bacterial circular DNA shit. And then that gets sequenced using an array of different things, including algorithms developed by NASA. This is a comparison of the various things. So the sequences can be anywhere from 36 base pairs long to thousands. And there's obviously price differences.
Quick question.
Yeah.
Which one did you use in like crime investigations? I'm just curious. You know?
I think that's actually just
Something way lower level.
yeah, and I've been meaning to look this up for years actually, because I haven't been sure what the hell they're talking about. But I think it's roughly either a restriction binding map, which is like there's various locations that bind certain enzymes, and you can map out where those are. If it's not that, then I feel it might just be something weird like a Western blot, which is, again, like just comparing like weights of proteins or product of some kind. I actually don't really know what a Western blot does. That's like genetics 101. I skipped to 400 level for that job and ignored all the shit that I should have known as the basics. And then learned it to give you this fine lecture. Yeah, so this is one of the sequencing technologies, actually two of them. Ion Torrent uses weird polarized balls to collect DNA and then goes into a little ball hole. And then like chunks of DNA get read off the end of like the ball hairs.
Nice.
All right, so it's really computationally expensive to have billions of short-ass fragments of DNA and then just try to make up what order they go in, which is what happens. So to cheat, or not really to cheat, but to do it less wrong, there's a reference sequence for major species such as human and mouse. And then there's a bunch of really shitty sequences for about everything else. The Human Genome Project is what made the reference sequence for humans. That was a whole big deal. It cost $1 billion. It's now about $10,000 to sequence a human. But that's because they had to do it really right the first time around. So the reference sequence is just based on the premise that people are very closely related, the whole 0.1 to 0.5% difference. That once you have a sequence of one person, it's real easy to then take these chunks and see where they match up to other people. And it is pretty easy except for that whole crap about repeats. There's tons of places where your little chunk is going to look like exactly the same here as over here, and you can't really tell which of those repeats that little chunk is supposed to go to. So then you get crap like this, where it's just all these chunks of DNA that get mapped to the reference.
Who was the original reference?
There's two original references. So while the Human Genome Project was going on, that was an NIH-funded thing, and I forget the main guy in that, and that was the really expensive one. There was also this guy, Craig Venter, who was just like, fuck it, I'm going to sequence my damn self. I've got tons of money. And actually used this technique without a reference to show that it could be done, where he, using longer reads than are usually done here, he just did the same thing but constructed everything from scratch. For the Human Genome Project, what they did was they broke it up into specific, they first made libraries of each chromosome that broke that up into specific sections. And then eventually they got to this point, but they broke it up into chunks so that they knew at least roughly where their chunks were going first. And Craig Venter was just like, fuck that, computers. We can totally just use computers. And it worked. It came out with almost the same quality sequence. The guy also regards DNA as a purely a programming language and thinks that we can just program anything we want with it. Kind of. And then, yeah, so this is how, so then it lines up all these chunks of DNA, and then when enough of them line up that look really good, except for like certain problems, that's when it says there's, hey, a mutation here, because all of the rest of this is the exact same. But this doesn't match the reference. So this is a mutation, or a variation, because there's no saying that that one isn't the more common variant, and that the person who is the reference actually had a mutation there that was them specific. All right, so there's a lot of problems with this. Although
Errors?
yes, you like that? So there's several errors that can arise from this. What's amazing is that there are these errors, and there's a lot of shitty research that doesn't compensate for them properly. But for the most part, this shit actually works pretty goddamn well. So I already mentioned reads can match multiple areas because of the whole repeats thing. Sometimes this results in mutations being called some place that they're not. Also with certain kinds of certain kinds of sequence, it matters how many reads you get in a location, because that's telling you how expressed a gene is. So it can tell you the wrong expression level for a gene. Sometimes it won't match at all because these machines are they've still got sequencing errors. The machines are imperfect technologies. And yeah. So on to epigenetics. Gene silencing, gene activation. Genes can be turned on and off. So now you got the whole DNA. That is your Mendelian fucking backbone of all this shit. Now you can tell if someone has a gene. But the epigenetics is how they get turned on and off, besides them just being mutationally fucked so they don't work. They can also have this. So there's various controls. Not all cells will express all genes, because then they'd all be the same cell, and then we'd all be made out of a stem cell group. And wouldn't that be delightful? So yeah, that's how it breaks it up into the different cell lines based on these markers. So going back to gene transcription, you've got your, over here, this is your gene over here. And then there's a whole bunch of stuff that's upstream of it that gets read first. You've got your promoters, you've got your enhancers, you've got insulators and silencers and various things. And all of these are controlled by these epigenetic factors that can turn the genes on and off. So based on like methylation of your activating assembly, this vacuum cleaner here may or may not might not actually read the gene. You've got there's various, yeah. This might inhibit it. This might help it work a little bit. It's not a linear function. It's part of the problem, or not really a problem. It's part of the difficulty in figuring it all out. I assume you can all read that. All right. So that whole chromatin thing that I mentioned before. So histones are these There's actually 4 histones in each one of these little balls. DNA wraps around them every 140 base pairs, and as best I know, it's exactly 140 every time, but I could be wrong. What do these histones do? Well, you've got a whole mess of bubbles. All right, so you've got the 4 different histones, and basically based on what's there as far as methylation, acetylation, ubiquitination, propylation, phosphorylation, I made up propylation. This will cause the DNA to be more tightly packed or more loosely packed. The more loosely packed it is, the easier it is to read. Generally speaking. So, basically along the same lines as what I showed before, but this is with a lot more bubbles. So you've got down there the promoter region, the enhancer, if it's got the right methylation patterns or whatever, folds over, helps the promoter promote, which will then fold over, do something which will help the gene activate using gene activation protocol. Then you've got DNA methylation, which is different from histone methylation. This is also where I was talking about there's 4 bases except then there's 5 because you've got CGAT, but then you've also got methyl C, and then you've also got hydroxymethyl C. Methylation in the promoter region actually turns off genes. Methylation in the gene region helps turn on genes. And this methylation tends to happen in regions of high CG content for reasons that people don't really understand. But it happens around what's called CPG islands. It actually happens on CPG island shores. So vertebrate genomes, for some reason that no one really fucking knows, have a depleted level of CG content. It's not like 25% of each nucleotide. The CG content actually is like, I think 40% in vertebrate species or something around that. The CPG regions are defined as greater than 60%. They tend to be about that long. They exist in those kind of regions of promoters. But yeah.
How about plants? Do you know if there's CPG?
Fuck plants. Plants are a fucking mess, and I'm not touching them. First off, I don't know much about them. Also, all that shit I said about repeats. Plants are just huge chunks of DNA that repeat themselves over and over again into huge monstrous genomes. The bioinformaticist in my lab that actually knows what the fuck he's doing, his big thing before coming to the lab was doing the genetic sequence of rice, which apparently is a giant fucking mess. So yeah, DNA methylation. Look at that. See? See, it silences. Except when it helps transcription up there. We're learning. Alright, so DNA methylation can turn things on and off at various areas of the cell, and it's actually those of us with 2 X chromosomes. It's through DNA methylation that they get turned on and off. Actually, if you have both X's active, you're fucked. It leads to all kinds of disorders. So usually one of those X's is turned off, but what's crazy about all that is it depends on the tissue and depends on the cell. So this is a rat brain. On the left is only paternally inherited X chromosome. On the right is only maternally inherited X chromosome. These are retinas, or irises rather, and you can see a similar pattern, although not as clear-cut, but where it's almost entirely one or the other X depending on the retina. And then these are other tissues that are also showing weird shit.
I got a quick question.
Yeah.
Did they make a rat with only the paternally inherited on one side, or is that?
No, that's just what fucking happens. Like, if you've got 2 X's, you can't have them both on or you have problems. Yeah. But for some reason, which one is the active one depends on which tissue you're in, or sometimes which cell within the tissue. And it's crazy like that. This happens partly through what's called imprinting. So the DNA methylation imprints on the germ-line cells, i.e. the ovary and sperm. And then this can turn on and off genes that then get passed down. In this example here, actually the one that got turned off is the one that would prevent cancer. I don't know what was the first image on Google. But oftentimes actually this imprinting is generally regarded as a beneficial thing, and one where it's more likely that the more useful of two will get turned on. So besides just the whole X chromosome, a lot of your genes can be turned on and off on an individual basis by your parents. So during embryonic development, egg gets released, hangs out, gets completely demethylated. All of it just gets stripped away. Then during mitotic rest, I don't remember cell cycle for shit. I don't know what the fuck that means. It gets de novo remethylated. Oh wait, I'm sorry, that's the sperm. This is the egg. Sperm earlier on gets de novo remethylated. The egg actually gets de novo remethylated up here. So then you've got the zygote after they hang out for a minute. And it gets almost entirely demethylated on the paternal and paternal side, except for those regions that were imprinted maternally or paternally, which just sort of hang out. I also saw a researcher give a talk who claimed that using these, by using DNA methylation techniques, they were able to degender rats. So yeah, here's the environment can affect gene expression, and then that can be passed down.
Nalgene bottles.
Huh?
Bisphenol A, that was in Nalgene bottles.
Yeah, yeah. So these rats, first off, this is wrong. These two mice are not genetically identical because no one's genetically identical, but they're pretty goddamn close. But yeah, they were able to do experiments like this. There's a ton of experiments, and also social experiments have been done, like stress in minors having passed down something. I don't fucking remember now that I'm talking about it. Anyway, don't be a stressed out minor. It might affect your grandchildren. You'll just have to take my word on that one. By minor, I mean like a minor in West Virginia, not a stressed out child. Don't be a stressed out child either. You're all too old for that. So then you've got chimerism and mosaicism, which get constantly conflated in the literature, and I just put them in here because I wanted to put them in here. Because chimerism is, it's sort of like what I was talking about with the different genes and chromosomes being turned on and off, except you can have two genetically distinct cells. Sometimes this happens when you have fraternal twins and one of them basically eats the other before they get to like any phase where the word eating would be accurate. But they basically combine into one thing, and then you have a child or an offspring that actually has two whole genotypes. And then the different genotypes get expressed differently in different cells. And then you've got mosaicism, which is kind of the same thing. Chimerism can be cool though. It also leads to people with like different colored eyes sometimes. Mosaicism, that was the first image that came up, and all the other images look kind of like that, so I just didn't put it in a real slide. All right, so gene expression, what we've been kind of talking about here with the epigenetics. So it's not just an on and off thing though, it is variable. So all these different colors are like how much a gene shows up based on all the different effects I've been talking about. And then you get these, and then the dendrograms up top, which show how much things cluster, which you then use to say there's an effect between a thing, another thing. But affecting one gene doesn't just affect one gene often. It affects another gene, which affects another gene, which will then affect another gene, it might affect the first gene. So that's another reason it's not binary, and another reason that everything gets crazy, because you've got pathways. So when you affect one of those genes, you can affect a ton of other genes. This is pathways in cancer. This one, what? Oh, that's Huntington's disease. It's a good one. Oh, the gap junction. If you're into gap junctions, that's a pathway for that. Also, a lot of these pathways are horribly inaccurate because it's really hard to figure out what the fuck's going on. And some of the ontology and pathways are determined basically by scientists being like, I'm going to stick this protein next to this protein in a dish and see if they like make out. And if they make out, then they say they're related in some way. Don't you make out with anyone you're related to, you're not a gene. Alright, I'm getting towards the end here. So you know, Connor.
Thank you.
Genetic alteration. This is very brief because I know actually kind of jack shit about it. There's various ways you can do this. You can do gene targeting, which is sort of the way where you just inject a germ cell line, so an egg and sperm with something packing the DNA that you want, usually using like a virus as vector to shove the DNA in there, or you can use the DNA from another animal, which would be like cloning. So you do that to an egg, you do that to a sperm, and then you breed them together until you got the pure mutant that you want.
Is that how you make like really dank kush?
Yes. That is exactly how I make it, but not everyone has access to the technologies that I do. You can learn all about it in this fun-filled pamphlet entitled Grow Me by Life Technologies.
Hey, you give that to me.
Yes. I'm an asshole. All right, so another way. Nuclease sequence modification. Remember all that crap about like how DNA fixes itself and how it recombines in chromosomes and all that? Well, scientists were like, well, if DNA can do that, why don't we just use DNA to do what DNA does real bad on itself in ways that let us modify shit? And that's what they did with that. It's like, hey, we're going to take all the proteins and enzymes and shit that like make DNA break itself apart, put itself back together in different configurations, and use it to do it the way that we want. Which is also how CRISPR works, which is a different thing that we're using in our lab, but I don't know jack shit about either. But I brought it up because at least I know more about it than the other two things I should. But look, it like cuts a piece out and then shoves a chunk in. There are issues with things like this. Basically, again, this is a technique that can be used on a live organism. Basically, if you give it enough of a viral pack to just sort of have viri floating around your body to shove extra DNA in there as necessary. There's problems with that, of course, because you need enough to make sure it infects all cells, which isn't always going to work. Yeah. Cloning. Birth to cow poops, eggs, and then you got another cow. All right, metagenomics. So we've been through genomics and epigenomics. Let's get to metagenomics. It's short, it's easy. All right, so we have 10 trillion human cells in our bodies and 100 trillion bacterial cells. They interact